Welcome to AFRbase browsing tutorial. We are glad that you're here. This is the home page of our database. And as you can see, AFRbase, that is antifungal resistance database, majorly consists of 4 panels on the home page. This is the menu panel. On the left side, you can see the browsing categories. On the right panel, you can see the stats of the database, that is AFRbase in numbers. And in the central panel, you can see the objectives with which this database has been compiled. This is a compilation of mutations in fungal proteins which are responsible for conferring resistance against known antifungal drugs. And this data stats is of host-wise mutation distribution, pathogen-wise mutation distribution, and drug-wise mutation distribution. As you can see, we have covered usually 3 hosts: animal, human, and plants. These are the fungal pathogens covered, and these are the drug classes which have been covered in this database. So now, moving on to the search page of this database, there are 3 methods through which we can search data in AFRbase. One is advanced search, keyword-based search, or searching the complete AFRbase data. For advanced search, if you know the gene or the drug or the pathogen you are searching for, then you can go for advanced search. For example, if you know the name of the gene for which you want to see the mutations, click on the Browse by Gene page. From the list of genes, you can select the gene of your interest and simply click the Submit button. Here you can see the mutations recorded for the gene selected will appear, and along with the drug against which the resistance is conferred by that particular mutation for all the fungal pathogens. If you want to know the details, if you want to know the details of a particular ID, you can simply click on that ID, and as you can see, the additional details compiled through literature, that is disease caused, mode of transmission, gene locus, are available in this page. And also you can find links to different PubMed IDs, that is of the literature articles from which this mutation has been retrieved, are available for more information. Similarly, if you know the name of the drug you are searching for, you can simply click on the Browse by Drug button, choose the drug of your interest, and you can search mutations. And similarly, they are sorted by pathogen. If you know the name of the pathogen for which you want to see the mutations, kindly click here and click the Submit button. Now moving on to the second part of search, keyword-based search can be done if you have any specific keyword in mind. Examples have been provided here. You can simply type in this bar and click on the search button. All the entries containing your keyword will appear as we searched for host animal. All the entries with animal host appeared in the search results. Then again, if you want to see the complete data of AFRbase, simply click on View All Data and press the Submit button. Here, there is another option to sort the research results in ascending or descending order based on the mutation count per gene. That is, if you sort the data by ascending, the entries with least number of mutations will appear first. Similarly, you can do for descending. This will be helpful if you are trying to search a gene with least or a maximum number of mutations recorded in this database. Then after search, we have the BLAST tab. If you want to perform a BLAST search of your query against the data in this database, you can simply enter your query here or upload a FASTA file through this button. For example, if we enter the query here, please make sure that you enter it in FASTA format beginning with a less than sign, and you can choose the E value from this drop down and the matrix value, that is Blossom or PAM, from this drop down, and click on the submit button. Blast results sorted by E value appear here, and if you want to know more details about your top hit, you can simply click on the UniProt ID and it will take you to the UniProt results of this particular protein that is your top hit. So after the blast tab, we have the 2D and 3D view tabs. So if you want to view the mutations of a particular gene in a holistic fashion, there are two methods, two ways, 2D view or 3D view. In 2D view, we have constructed two-dimensional mutation hotspots of every gene. You can simply click on the relevant gene button and see the mutation hotspot, a two-dimensional plot of all the mutations recorded for a particular gene. On the x-axis, you can see the length of the protein, that is 528 amino acids, and on the y-axis, you can see that particular mutation recorded, as in on 33rd position, if Y is substituted by C, it will cause a mutation, and that mutation will cause resistance against fluconazole, itraconazole, and voriconazole. So that is the color of these lollipops indicate the drug against which resistance is being caused, and these positions are the mutations which confer this resistance. Similarly, you can see for other genes. In the 3D view tab, we have installed JS Small plugin to have a look at the mutations in 3D view. That is, we have converted the PDB coordinates to the UniProt coordinates, and if you wish to see the gene of your interest in 3D, you can visit this tab. That is, you simply click here to activate the JS Small plugin. The application is loaded and you can rotate and follow different options available here. To have emphasized view, you can change the background color as per you desire. You can turn the spin on or off option, and there are various other options available with the JS Small plugin. Herein, as you can see, all the regions colored red indicate that those regions confer mutation against this particular drug, that is fluconazole, independently as well as together. So this is all about the 3D view of mutations in AFRbase. Other than that, if you encounter a mutation which has not been recorded in this database or have sequenced one, we would request you to please make a entry in the mutation submission form. That is, you can submit your entries through this form and we will be happy to incorporate in the database after curation. To see the stats of the data contained in this database, this is the statistics tab where we have categorized gene-wise, drug-wise, and pathogen-wise mutations, and the others have been tabulated here. If you have any queries related to our database, we'll be happy to answer them through these email IDs provided here. And the help page, as you already know, contains the video tutorials of browsing through this database. Thank you, and we hope this database will be helpful in your research.